Welcome to class, student. Today we'll be looking at a very important topic titled Enzymes. Introduction. Enzymes are biological catalysts, primarily proteins that accelerate chemical reaction within living organisms. They are essential for various life processes such as digestion, building muscle, and destroying toxins. Without enzymes, this reaction would occur far too slowly to sustain life. Key components of enzymes are biological catalysts, proteins, specific activities essential for life regulations. Biological catalysts, proteins, specific activity essential for life and biological catalyst is one of the components of enzymes. Enzymes are catalysts, meaning they speed up chemical reactions without being consumed in the process. Protein mostly. While some enzymes can be RNA, the vast majority are proteins, complex molecule made up of amino acids. So majority of enzymes are proteinous in nature, while some are RNA and they are complex molecules that are made up of amino acids. Specific activity. Each enzyme typically catalyzes a specific reaction acting on particular molecule called substrate. Essential for life, enzymes are crucial for a wide range of bodily functions, including breaking down food, DNA replication, and building muscle. Examples are of enzymes are digestive enzymes in the stomach and intestine help break down food into smaller molecules. So we have examples are the digestive enzymes in the stomach and in the intestine, and their function is to break down food into smaller molecules. We want to look at regulation. Enzymes can be regulated by various factors including temperature, pH, and the presence of cofactors, non-protein helpers. Enzymes are specialized proteins that govern virtually every complex biochemical processes occurring in all living organisms, from microscopic microorganisms to complex animals and plants. These remarkable molecular machines demonstrate extraordinary efficiency and precision, accelerating specific chemical reactions far more effectively than any artificial catalyst created by humans. What makes enzymes particularly fascinating is their selectivity. Each enzyme typically catalyzes only one specific type of chemical reaction involving one particular type of compound. Their activity is carefully controlled by regulatory molecules called activators and inhibitors, which can either initiate or halt enzymatic reaction as needed. The sheer abundance of enzymes in living systems is staggering. Every cell contains these catalytic proteins, though their types and quantities vary according to cellular function. To put this in perspective, an average mammalian cell measuring roughly 1 billionth the size of single water droplet houses approximately 3,000 different enzymes working in concert to maintain life. Enzymatic function in living system. Enzymes serves multiple essential roles that make life possible. We have the digestive processes, the biosynthetic activity, and vital life function. The digestive processes, biosynthetic activities, vital life functions. So, digestive processes. Certain enzymes specialize in dismantling large nutrient molecules, proteins, fats, and carbohydrates into smaller more manageable components during digestion in the stomach and intestine. Additional enzymes facilitate the transport of these broken down molecules through intestinal walls to the bloodstream. Biosynthetic activity: other enzymes work in the opposite direction, assembling small simple molecules into larger complex structure required for cellular components and tissues. Vital life function: enzymes orchestrate energy storage and release reproductive processes, respiration, and even function, and also vision. So vital functions of enzymes: they orchestrate energy storage and release. They also work on reproductive processes, respiration, and even vision. Without this molecular catalyst, life as we understand would cease to exist. Disease and enzyme deficiency: when enzymes malfunction or are absent, serious health consequences can result. Using our pathway example, if compound A must be converted to product E through series of enzymatic steps, but one enzyme in the chain is defective, the final product E may not form properly. If E is essential for vital body function, disease may occur or disease occurs. Many inherited human disorders stem from single enzyme deficiencies. Albinism exemplifies this relationship. It results from the inherited inability to produce tyrosine, an enzyme required for synthesizing the pigment that give color to hair and eyes. The absence of this single enzyme disrupts the entire pigmentation pathway, leading to the complete absence of melanin production. Medical and industrial application: modern medicine increasingly relies on the therapeutic enzymes. Thrombin accelerates wound healing, while the other enzymes diagnose disease, treat certain leukemia types, and counteract dangerous allergic reaction to penicillin. Lysozymes serve as a natural antibiotic by destroying bacterial cell walls, and researchers continue exploring enzymatic treatment for tooth decay prevention and blood clot solution. Beyond healthcare, enzymes drive numerous industrial processes. Food production depends heavily on enzymatic reaction, from cheese manufacturing and beer brewing to wine aging and bread baking. Industrial applications span leather tanning, chemical synthesis, and laundry detergent formulation. Enzymes also function as sensitive analytical tools, detecting minute quantities of specific substance in laboratory testing. The six category classification system are oxidoreductases, that is hydrogen transfer, hydrolysis, water addition, transferesis, non-hydrogen substance transfer, lysis, isomeresis, and ligases. Oxidoreductases: hydrogen transfer, hydrolysis, water addition, transferesis, non-hydrogen substance transfer, lysis, isomeresis, and ligases. The oxidoreductases facilitate reaction involving hydrogen transfer between molecules. Hydrolysis catalyzes addition of water components at specific molecular locations. Transferesis manage the movement of substance other than hydrogen between different molecules. The remaining three categories, lysis, isomeresis, and ligases, each handle specialized reaction type with unique characteristics. Remarkably, oxidoreductases and transferesis together comprise approximately half of the roughly 1,000 enzymes currently recognized and catalogued by researchers worldwide. The enzymes identified with hereditary disease: we have disease name and defective enzyme. Albinism: the defective enzyme is tyrosinase, while phenylketonuria is phenylalanine hydrolase. phenylalanine hydroxylase. That is the defective enzyme. We have the photosuria. The defective enzyme is fructokinase. While we have the galactosemia. The defective enzyme is galactose 1 phosphate iode transferase. We also have the methemoglobinemia. Methemoglobinemia. The disease is methemoglobin reductase. Let me go take that again. We have methemoglobinemia. And the defective enzyme is methemoglobin reductase. Other functions are therapeutic and medical application. The medical field has embraced enzymes as powerful therapeutic tools with expanding application. Thrombin serves as a wound healing accelerator, helping injured tissue repair more efficiently. Medical professionals now employ various enzymes for disease diagnosis, including remission in specific leukemia types that affect blood-forming organs and neutralizing dangerous allergic responses in patients sensitive to penicillin. Lysozyme function as a neutral antimicrobial agent by breaking down bacterial cell walls efficiently or effectively destroying harmful microorganisms. Researchers continue exploring enzymatic solutions for preventing dental cavities and developing anticoagulant treatment for thrombosis, a condition where dangerous blood clots form within blood vessels. Future medical applications may include targeted therapies for correcting enzyme deficiencies and abnormalities associated with various diseases. Industrial and chemical use. Enzymes have revolutionized numerous industrial sectors through their precise catalytic abilities. Manufacturing process utilizes enzymes for synthesizing specific chemical compounds and processing leader through tanning procedures. Their exceptional sensitivity makes them invaluable in analytical application where they can detect extraordinarily small amounts of particular substance with remarkable accuracy. The food industry depends heavily on enzymatic process across multiple sectors. Cheese production, beer brewing, wine maturation and bread baking all rely on specific enzymes to achieve desired flavor, texture and chemical transformation. Even household applications have benefited from enzyme technology, particularly in laundry detergent designed to break down various stains and soil types. Thank you for listening.